Children, we discussed the two types of ribosomes. So according to the cell type. So if the cell types are two, we have two types of cell types. We have prokaryotic cells and we have another type of cell which is eukaryotic cell. And both the cells have different types of ribosomes. As I told you yesterday, the prokaryotic cells have 70S type of ribosomes and the eukaryotic cells have 80S type of ribosomes. S here is Swedenborg's unit. Or also known as sedimentation coefficient. Or sedimentation coefficient. Now, this S or sedimentation unit or a Swedenborg's unit or sedimentation coefficient, this is a non-SI unit. This is a non-SI unit. This is a non-SI unit. And along with this is measure of time. This is measure of time. And the value of this is S is equal to 1 into 10 raised to power minus 13 seconds. This is the value of S. So S is 1 into 10 raised to power minus 13 seconds. Now what is this basically? This is the rate at which this is the rate at which the ribosomes, we are talking here about ribosomes, so ribosomes settle at the bottom of centrifuge tube when centrifuged. That means when you take them in a test tube, when these are taken in a test tube and then rotated in a centrifuge machine, these test tubes are inserted in these circles and these are rotated at a very high speed. What happens? They settle at the bottom. Now the rate at which they settle at the bottom of the centrifuge tube, this is known as Swedenborg's unit. Now when we say that it is 70S type, now when we say that in prokaryotes, it is 70S type. That means this ribosome all together. See, ribosome, when we say ribosome, what does a ribosome means? A ribosome is, it consists of a larger subunit, and a smaller subunit. Ribosome consists of this. That means that this is a smaller subunit and this is the larger subunit, and together it makes a ribosome. This is one ribosome, but they are not attached to each other all the time. They are not attached to each other like this all the time. In the cell, in the cytoplasm, they lie free. This is smaller lying somewhere, the larger lying attached to the ribosome. Mostly the larger subunit, it lies attached to the ribosome. Here, this is ribosome. So they lie attached to the ribosome, the larger ones. And the smaller ones, they lie here and there in the cytoplasm. They come together only when they perform their function. They attach to each other or bind with each other only when they perform their function. Now, I'm telling you, ribosome is together as a whole. When we say it is a ribosome, it consists of a larger subunit and a smaller subunit. So when we say in prokaryotes, the ribosome is 80 I'm saying here that the by this subunit and a larger subunit. It will have a smaller subunit and a larger subunit, and together it will make 70S. Now, when I say sedimentation rate of the ribosome in prokaryotic cell is 70S, it will have a larger subunit and a smaller subunit. Now, the S of the smaller subunit and the larger subunit will be different. In case of prokaryotic cell, the S of the in 70S, it is the smaller subunit is 30S. This is 30S, and the larger subunit is 60S. Now, this is something to be considered, that together the ribosome is 70S, but ribosome consists of a smaller subunit and a larger subunit. In the 70S ribosome, the smaller subunit is 36, 30S, and the larger subunit is 60S. What do we mean by this? If together this is centrifuged as a whole, the complete ribosome is centrifuged. I'm making the complete ribosome of 70S type. This is the smaller subunit. This is the larger subunit. This is 70S. When this is centrifuged as a whole, when this is centrifuged as a whole together, then its Sweetberg's unit is 70S. That means the Sweetberg's unit in this case is 70S. That means when this total ribosome in the binding state, when it is centrifuged in a centrifuge machine, it settles down. It settles down at the bottom of the test tube in 70S time. That is why we say that in prokaryotic cell, the ribosome is 70S time, because as a whole, in bound condition, when the larger and the smaller subunits are bind, when they are attached to each other, their Sweetberg's unit or their settling time is 70S. Now, if we take only the smaller subunit of this ribosome, which is found in prokaryotic cell, the smaller subunit in prokaryotic cell, I'm talking here about prokaryotic cell, smaller subunit has a Sweetberg's time or sedimentation coefficient of 30S. That means the smaller subunit, when centrifuged in a centrifuge machine, it settles down at 30S. And the larger subunit in a prokaryotic cell, larger subunit is 60S type. It is 60S. That means this settles in the centrifuge machine at this time. This is the sedimentation coefficient. If the larger sediment be 60S, that means it's settling. 70S type is found in prokaryotic cell. So is it clear to you, this prokaryotic cell 70S type? Is it clear, beta? Any query in this? Anything you could not understand in this? So that I move on to the next one, which is 80S type. Is it clear in this? Any question? Okay, Ashika is saying that it's clear. Yes. Okay, fine. This is something important and you should know it, because when you see the prokaryotic cell have 70S, smaller subunit 30S, larger subunit 60S, you will not be able to understand unless you come to know that this is the settling time. This is the time taken to settle. And I'll write this for eukaryotic cell over here. So, as I told you, the ribosomes in eukaryotic cells, that means the cells in which the nucleus is very clear. So, in those, the ribosomes are 80S type. What do we mean by this? This means the settling time of ribosome as a whole. That means smaller, larger subunit and smaller subunit. When they are bind together as a whole, the Sweetberg's unit or the settling time in the centrifuge machine is ATS as a whole. But it also has two subunits, a smaller subunit. I'm writing just, okay, I'll write full so that you understand. Smaller subunit and a larger subunit. This is the larger subunit, and this is the smaller subunit. Okay, so it has both. When together in binding state, here they are in binding state. That means the larger subunit and the smaller subunit attached to each other. When it is centrifuged, then in that case the sedimentation coefficient is ATS. Now, if these are separated, because I told you that they join together only when they function. Otherwise, most of the time, both the subunits, the smaller subunit and the larger subunit, they remain separated in the cell. Larger subunit mostly remain attached to endoplasmic reticulum and the nucleus, whereas the smaller subunits, they remain free in the cytoplasm. So mostly they remain free, in detached state. That means both the subunits separated. But when they perform their function at that time, both of them, and they come and bind together, and when as a whole both the subunits, their Sweetberg constant or their sedimentation coefficient is taken, it comes to be ATS type. Now, when they separate, and most of the time they are separated except for when they function, when the smaller subunit, its Sweetberg constant or sedimentation coefficient is taken, it comes to be 40S. 40S is its sedimentation coefficient, whereas the larger subunit, its sedimentation coefficient comes to be 80S. No, not 80S, 70S. Just a minute. Yes, it comes to be, this is beta 50S. This is not 60S. I've written here wrong. This is 50S. This is 60S. It comes to be 60S in case of larger subunit. So if it is a eukaryotic cell, it will be 80S as a whole when both the subunits, they are in binding state. But when they separate, as most of the time they separate, they remain in the cytoplasm, in the on the nucleus, on the membrane of the nucleus, or on the membrane of the endoplasmic reticulum. So when they are in separated condition, if they are centrifuged, the smaller subunit, its sedimentation coefficient is 40S, and the larger subunit, its sedimentation coefficient is 60S. So this is about the eukaryotic cell. Any question or anything you want me to repeat? Yes, beta, anything in this you want me to repeat, or you could not understand? Sedimentation coefficient is very important, and you can be asked a question, what is the sedimentation coefficient in both the cell types? When I say cell types, that means I'm talking about prokaryotic cells and eukaryotic cells. So you will have to mention that in eukaryotic cell, it is ATS. In binding state, it is ATS. The sedimentation coefficient is ATS. But when both the subunits, they are separated, as mostly they remain separated, the smaller ones remain in the cytoplasm, the larger subunit, it remains attached to the wall of the nucleus or the membrane of the endoplasmic reticulum. So in separated state, the smaller subunit is 40S and the larger subunit is 60S. So and about prokaryotic also, you will have to mention that together if they are taken, it is 70S, but separately they are this. Okay, so children, here any query in this, anything which you want to understand, or which you want me to repeat, which you could not understand? Yes, beta. Anything which you could not understand, you want me to repeat? Yes, bache. Any query in this? Or I should proceed with the next, which is the last topic of the ribosome. Okay, Rohan is saying it's clear. Okay, and others? I am getting just one response. Yes, beta. Is it clear to everybody? I'm not getting any response. I'm not getting any response, bache. Okay, Amrita, Yashika, Harsha, Ankita. Okay, Ankita. Others also. Siksha, Tadarani, fine. Yes. Lalit, is it clear? Sanvi, Kushman, Jasleen, Harsha, Sanil, is it clear to everybody? Okay, fine, fine. So this was about the types, cell types, and the sedimentation coefficient of the subunits of ribosome and as a whole also. Now I move on to the another topic of ribosome, which is function, its function. What is its main function? What is the function of ribosomes? So I move to this topic, functions. You know what is another name of ribosome? If you know what is another name of ribosome, I think you all are unmute. You can speak up. You can unmute yourself and speak up. What is another name of ribosomes? Ribosomes are also known as, yes, bache, because the functions are based on that particular term, which tells what is the other name of ribosome. Yes. What are ribosomes also known as? Ribosomes are also known as, yes, who's going to tell me? Yes, Yashika. Pallid particles, yes, this was one of the name, and this name is because of the discoverer. Okay, he's the one who first observed it. Okay, because of that, these are known as pallid particles. This is because of the name of the researcher, because he gave the term pallid to the ribosomes to the, he gave the term ribosomes. And the name of the researcher was pallid. That is why on the name of this researcher, pallid, the ribosomes are also known as pallid particles. Now, one more term is there. If we consider the function of this, one more term is there, which is based on their function. What is that? Another name of ribosome. Anybody who can tell? What is that? Another name of ribosome? That is based on the function of ribosome. Yes, Amrita, Sanvi, protein synthesis, site of protein synthesis. Yes, this is in a sentence. Actually, this is not a term. Basically, what they are known as is protein factories. If you know, they are known as protein factories. Protein factories of the cell. They are known as protein factories of the cell. That means they are involved with protein synthesis. The main function is protein synthesis. Their function is protein synthesis. So protein synthesis takes place. This is their main function. Now, as I the endoplasmic reticulum also, this is roughly I've made endoplasmic reticulum. The smaller subunits, they remain free in the cytoplasm. These are the smaller subunits which I have made. They remain free. And the larger subunits, they remain attached. Mostly they remain attached to the nucleus and to the membrane of, first to the nuclear membrane and the membrane of endoplasmic reticulum like this. They remain attached here like this. So the larger subunit, they remain attached to these two membranes, whereas the smaller subunits, these are smaller subunits. And these are larger subunits, these also. Together, these are larger subunits. They are attached to these membranes. Mostly they remain detached from each other. Both the subunits, they remain detached. But when they perform this function, mostly detached, mostly in detached state, detached state, mostly in detached state. But when they perform their function, while performing the function, what is their function? Protein synthesis. cases. The smaller subunit and the larger subunit, they come and attach with each other. The smaller subunit and the larger subunit, they come and attach with each other. And this attachment is known as assembly. This attachment is known as assembly. When they attach to each other to perform the function, this attachment is known as assembly. Now, this attachment is possible only when the there in the cell magnesium ions are present. This assembly takes place in the presence of magnesium ion. So these magnesium ions should be present. They should be present. And what concentration? They should be present in 0.001 molar. Their concentration should be this much. When magnesium ions are present in this concentration, the smaller subunit, it attaches to the larger subunit, and this process of attachment is known as assembly. And the magnesium ion should be in this concentration. Now, if the magnesium ions, they are more than this, if these are more than 0.001, first I'll tell you less than. If these are less than, then what happens? If these are more than 0.001 molar, then what happens? Then what happens in that case? So when it is less than that, then no binding will take place. No binding. So what will happen? They will dissociate. Both the subunits will dissociation of both the subunits. Both the subunits. They will dissociate. They will separate. The small here, large here. Both the subunits will separate if it is less than this. If it is more than this, in this condition, what will happen? If this is more than this, then I think you remember when I was teaching you bacterial cell. If you remember, when I was teaching you bacterial cell, I made the bacterial cell like this, and in the center, I made little of this incipient nucleus. I made